Hello everyone. Today we're going to talk about the levels of protein structure, where I'll be emphasizing on the primary and secondary structure of a protein. I'm Ms. Shweta Sharma. I'm an assistant professor from Department of Biotechnology of Thakur College in Science and Commerce. So, proteins are the most abundant biological macromolecules which occur in all cells and all parts of the cells. They are one of the most interesting uh, biomolecules that we have. The reason for their why they are such interesting molecules is because there is a lot of structural as well as functional diversity of proteins. If you look at some of the proteins that we have, proteins form the hooves of cattle. They also form the horns of most of the animals. They are also present in the claws of birds. The spider's web, which is a very interesting molecule, which is characterized by its tensile strength and elasticity, is also a protein. And very interestingly, the silk obtained from silkworm is also a protein. All of these proteins that we can see over here, uh, they're very tough, rigid structures, and they provide strength to whatever structures they are present. And mostly when we find tough structures in nature, they're usually polysaccharides like cellulose. But here we can see that the toughest of the structures present over here are all made up of proteins. On the other hand, there are also proteins which are functionally very important. So if you look at the hemoglobin present in RBCs, where they help to transport oxygen, or if you look at dietary proteins like egg albumin, another dietary protein is casein, which is present in milk. And a very interesting protein, if you see the GFP, which is the green fluorescent protein, which is obtained from the jellyfish, and it is also used as a reporter gene in many microorganisms. So all of these are also more different types of proteins. Intracellularly, if you look, proteins have an extremely important role to play. So all enzymes are proteins, and we know that enzymes are very important for the functioning of the cell. Almost all cellular processes are carried out by the enzymes. So a few of them and their activities have been shown over here. Similarly, if we talk about the immunity of our body, immunoglobulins, which fight against the pathogens, are also protein in nature. And one very, very interesting type of a protein are the motor proteins which are present in our cell, which carry the cargo of various messengers, and they walk along the cytoskeleton track. And this track is also made up of proteins. So this motor protein, which is carrying the cargo, is also a protein, as well as the track is also a protein. So if you see at all of these different types of proteins, we can see that proteins have a lot of structural diversity, they have a lot of functional diversity. And that is why I started this session with saying that proteins are an extremely interesting group of molecules. So they are highly diverse group of biomolecules. There is a lot of structural diversity. Associated with the structural diversity, there is a lot of functional diversity. But surprisingly, all proteins are made up of the same 20 amino acids. So whether it is the toughest structure that you find in keratin, which is present in the hooves or the claws or the feathers, that is also made up of the same 20 amino acids, which make up hemoglobin, myoglobin, immunoglobulins, all the enzymes. So it is very interesting to understand this, that the same 20 alphabets that we have can give so much diversity in the protein structure. So to understand these amino acids better, before we go to understanding the structure of proteins, we need to understand the nature of these amino acids. So if you look at a general structure of an amino acid, it basically has a chiral carbon in the center, and there are 4 groups attached to it. You have the amino group, the carboxyl group, the H, and a R group. So because it is attached to 4 different groups, we call the central carbon, the C alpha carbon, as the chiral carbon. And it is this R group which basically differs in the 20 amino acids and which determines the nature of the amino acid present. So if you look at this chart, gives us the details of the structure of the amino acids, the types of amino acids, and also how they are classified. So if we look at this first group on the top, these are all aliphatic amino acids, and they are non-polar. So alanine, glycine, isoleucine, leucine, proline, and valine. They belong to the category of non-polar amino acids, which are aliphatic in nature. We also have three amino acids, which are aromatic in nature: phenylalanine, tryptophan, and tyrosine. Apart from that, another group of amino acids that we have are the negatively charged aspartic acid, aspartate, and glutamic acid, which we also call as glutamate. The positively charged amino acids are arginine, histidine, and lysine. The rest of the amino acids belong to the category of amino acids which are uh, polar, but they are not uncharged. So that brings us to serine, threonine, cysteine, methionine, asparagine, and glutamine. So in all, we have these 20 amino acids which differ in their polarity, which differ in their charges, and it is this basic property of amino acids which finally determines the structure of a protein. So coming to the main topic of levels of protein structure. So basically we have four levels of protein structure: the primary, secondary, tertiary, and quaternary. So if you look at the first level, which is basically the primary structure, it is just the string of amino acids which are joined together by the peptide bonds. There is no structural information present in the primary structure. This primary structure then folds into what we call as a secondary structure, which is a more localized structure. So this is an example of one of the secondary structures, which is the alpha helix. There are other secondary structures also present. The combination of all the secondary structures together form what we call as the tertiary structure. So here we can see that the alpha helix is forming one of the structures, and a combination of all of these will give us the tertiary structure. If a protein is having a single polypeptide chain, it usually has the last level of the protein structure is the tertiary structure. But if it is a multimeric protein, these polypeptide chains then combine with other polypeptide chains, and that the manner in which they combine the information with for combination is basically the quaternary structure. So this is the last level of structure. So a protein like myoglobin will have only the tertiary structure, whereas a protein like hemoglobin, which is made up of 4 subunits, will have a quaternary structure as well. So in this lecture, we'll be only focusing on the primary and the secondary structure of the proteins. This is another diagram which talks about the levels of the protein, that is the primary, secondary, tertiary, and quaternary. So we can see that the primary structure is basically a chain of amino acids which are joined by the peptide bonds, starting with methionine and ending with leucine for this particular chain of amino acids. These will then fold into the two primary or common secondary structures, which is the alpha helix and the beta sheet. These alpha helices and beta sheets will then combine to give us the tertiary structure. And finally, these tertiary structures of many of the polypeptides will come together to give us the final protein, if it is a multimeric protein. So here we can see that three monomeric units of the protein are coming together and associating with each other to give us the quaternary structure of the protein. If we talk about the primary structure, as mentioned before, it is a sequence of amino acids in a protein. The basic sequence of the amino acid in the protein is called its primary structure. And as I mentioned before also, that there is no structural information in the primary structure. It is just the sequence of the amino acids. So if we look at this diagram, we can see that this is one amino acid which is linked to another amino acid through this particular bond. And this bond is called as the peptide bond, which is formed between the between the carbonyl carbon and the nitrogen of the amino group of the next amino acid. The bond is formed by removal of one single water molecule, and therefore we call, since water is removed from this structure, these are not called as amino acids but as amino acid residues. So each of these amino acids is known as an amino acid residue, and we can see that they are all linked to each other by this particular bond, which is called as the peptide bond. The peptide bond is a covalent bond and a very strong bond which is formed during the process of translation. And since it's a covalent bond, it is also very difficult to break this bond. It cannot be broken down by changing the physical parameters, and we require enzymes like proteases to break these bonds. So the basic backbone of a protein is formed by these peptide bonds, which are extremely tough bonds. And this is how the entire string of amino acids is formed, starting with the first amino acid, and it can go up to 50, 100 amino acids to give a single polypeptide, what we call as a polypeptide chain. Every polypeptide chain will have an N terminal, as we can see over here. The N terminal is for the amino group which is present at one end, and then we have the C terminal, which is for the carboxyl group, which is at present at the other end. So the primary structure of a protein or a polypeptide, protein polypeptide, will always be polar in nature, because it will have an N terminal and a C terminal, which help in which will characterize the two ends of a polypeptide chain. So if we look at the peptide bond, this is also a very interesting bond, and it gives a lot of characteristics to the entire protein. So the 6 atoms of the peptide bond lie in a single chain, with the oxygen atom of the carbonyl group and the hydrogen atom of the amide nitrogen, which are trans to each other. So if we look at this particular structure over here, we can see that this is the peptide bond. So we have the 6 atoms which are present in the plane are the the carbonyl carbon, the nitrogen, the next carbonyl carbon, the C alpha carbon, and oxygen and hydrogen, which are in the trans state, which are trans to each other. So this is the main peptide bond. And here you can see that all of these are present in a single plane, which we call as the amide plane. Now, the peptide bond is a highly rigid bond, and it has a very unique characteristic. The peptide bonds, which are the CN bonds, they are not able to rotate freely. Like, every bond has a certain degree of rotation that can occur across a bond or around a bond. However, the CN bond are unable to rotate freely because they have a what we call as a partial double bond character. And because of this, the rotation is only permitted around the NC bond, that is this bond, and it is permitted around the CC alpha bond, which is this bond. We will come to the characteristics of these later. Let's just talk about how does the peptide bond have a double bond character. So over here we can see that this is how the peptide bond forms. Oxygen, being highly electronegative, will pull the electron cloud towards itself, and as a result, the electron cloud of nitrogen will be pulled towards the carbon. And this results in a partial negative charge on oxygen and a positive charge on nitrogen, giving a double bond character between C and N. Now, because of this double bond character, the C and N bond are not able to rotate freely, and therefore we have rotations which are allowed only around the NC bond and the CC alpha bond. And therefore we can imagine the primary structure of a protein to be a series of planes which can be rotated around these bonds. So these bonds which are present between C and C alpha, it is called as a psi angle, whereas the bond rotation is called as the psi angle, which basically determines the degree to which the bond can be rotated. And the NC alpha bond is called as the phi angle. These two angles are very important because they characterize how much a particular peptide chain can be bent. And there are permissible limits. There are limits to which these phi and psi angles can take their values. Theoretically, they can take any value between plus 180 to minus 180. That is, a complete 360-degree turn can be done by these. But we have steric hindrances which do not allow them to move or to rotate freely. So there are only a few rotations which are allowed around these bonds. So the phi angles and the psi angles have limited values that they can take, and it is through these rotations that a polypeptide chain can fold to give us an actual structure of the protein. So these when the primary structure, that is the phi and the psi angles, they take up certain values, that results in folding of the polypeptide chain, which basically results in the formation of the secondary structure of the proteins. So the secondary structure basically refers to the local conformation of some part of a polypeptide chain, and very few types of secondary structures are particularly stable, and that is the reason is because, as I mentioned before, that there are only certain values that the phi and psi angles can take. So, and only they are stable. So that is why, even though we have a lot of diversity in the primary structure of a protein, the secondary structure may not be that diverse. There are very few secondary structures that can be taken up by proteins. So the two most prominent secondary structures are the alpha helix and the beta conformation, which we'll be talking about in this session. So coming to the alpha helix first, if we start with one type of a secondary structure, that is the alpha helix. So it is basically a helical structure where the peptide bond backbone, the polypeptide backbone, is tightly wound around an imaginary axis drawn longitudinally through the middle of the helix, and the R groups of the amino acids protrude outwards from the helical backbone. So the helical twist of the alpha helix is basically right-handed. So if we look at this particular structure, we can see that the straight polypeptide chain has been wound around to give a helical structure, and this helical structure is basically right-handed, which means that if you take the right hand and point the thumb upwards, so the thumb basically is the axis, whereas the fingers which are bent, they show the direction of the fold or of the helix. So this is a right-handed helix. All alpha helices are right-handed. Some left-handed helices are seen in the super secondary structures in fibrous proteins like collagen. If we look at the alpha helix from the top, this is how it will appear. So you can see that it's a helical structure, and these structures which are protruding outwards are the R groups of the amino acid residues. So this is how an alpha helix will appear from the top. Now each helical turn includes around 3.6 amino acid residues. The reason we are not saying complete 4 or complete 3 residues is basically because there are complete 3 amino acid residues which are included in one turn, whereas the first and the fourth amino acid, they are hydrogen bonded, which results in one third of the two amino acids, that is the first amino acid and the fourth amino acid, they have one third of themselves in the turn. So this results in the total value of around 3.6 amino acids per residues. The phi and psi angles which can be taken for the alpha helix to form. So psi angles can range between minus 45 degrees to 50 degrees, whereas the phi angles are usually around minus 60 degrees. So when these values are taken, the primary structure of a protein will usually fold up into an alpha helix. Now, the reason why this entire structure remains stable, like it will not fall apart and it will still remain as a helix, is because of extensive hydrogen bonding. And the hydrogen bonding takes place between the oxygen of one of the amino acids and the hydrogen of another amino acid. These two amino acids are usually placed, or they are placed around 4 amino acids away from each other. So the first amino acid and the fifth amino acid, if we consider the first amino acid as I, then I plus 4 would be the fifth amino acid, and these two would be linked to each other through a hydrogen bond. Similarly, the second and the sixth would be linked to each other through a hydrogen bond. The third and the seventh would be linked to each other as a hydrogen through a hydrogen bond, and so on and so forth. So if we look at the structure over here, we can see that this is the first amino acid, then the second amino acid, third amino acid, fourth amino acid, and this is the fifth amino acid. So we can see that the oxygen of the carbonyl carbon of the I amino acid is linked to the hydrogen of the amino group of the I plus 4 amino acid. And like that, this entire structure gets stabilized. So the reason why the alpha helix will remain as an alpha helix is because of this extensive hydrogen bonding. There are many other elements which also play a role. Apart from the hydrogen bonding, there are other elements which will also help in stabilizing the structure of the amino acid or stabilizing the structure of the alpha helix. And one of the most important factors which play a role in a polypeptide chain to fold up as a helix is the sequence and the identity of the amino acids which are there in that segment. So some amino acid residues, some sequences have a natural tendency to fold up as a alpha helix, whereas others might not be permitted to fold up as an alpha helix. So the tendency to fold up as a helix will depend upon the sequence of the amino acids. So let us look at a few examples that which where an alpha helix may not form, or where there is a higher tendency of an alpha helix to form. So if we look at negatively charged amino acids like glutamine residues. So in an alpha helix, you will not have two negatively charged amino acids placed next to each other, or you will not have two positively charged amino acids placed next to each other, because their R groups are going to repel each other. So if their R groups repel each other, they will prevent the formation of a helix. So you cannot place two negatively charged or two positively charged amino acids next to each other. They will not have a tendency to form a helix. So similarly, positively charged will not be placed next to each other, but there's a very high chance that a negatively charged amino acid would be 3 residues away from a positively charged amino acid, or vice versa. The reason for that is because if we have a positively charged amino acid 3 residues away from a negatively charged amino acids, they can permit the formation of an ion pair. So not only there is hydrogen bonding between the two amino acids, but there would also be a ionic bond formation. So that further stabilizes the entire structure of the alpha helix. Similarly, two aromatic amino acids will never be placed next to each other, and they would be usually spaced 3 residues away from each other. Uh, as a result of this, you can have a hydrophobic interaction, which will again help to stabilize the entire structure. So instead of placing these amino acids close to each other, if we allow them to be placed 3 amino acids away from each other, we can get a much more stable alpha helix. There are 2 amino acids which we usually do not find in an alpha helix, and they are proline and glycine. And the reasons why we might not find these 2 amino acids is completely opposite. One is extremely flexible, whereas one might not give the option of forming a hydrogen bond. So if we look at proline, the nitrogen atom in the proline is part of a imino group, and therefore the rotation about the NC bond is not possible. And therefore, proline is usually known as a helix breaker, and it is usually present at the end of an alpha helix. So proline, because of its rigid ring and because of inability of the rotation around the NC bond, we will not find this particular amino acid within an alpha helix. Glycine, on the other hand, is not found in the alpha helices because it is highly flexible. It has much more higher flexibility. Being the smallest amino acid, it can be very easily wound, and therefore the level of winding that we need for alpha helix may not be present, which may not be possible with glycine. And therefore both proline and glycine are the amino acids which are usually not found in an alpha helix. Apart from that, all positively charged, negatively charged non-polar amino acids will be found in an alpha helix, but they would usually be placed in such a way that allows for another non-covalent bond formation apart from hydrogen. So the entire structure of an alpha helix is basically stabilized by non-covalent interactions, whereas the main structure is formed by the strong peptide bond, which has been wound around in an alpha helix. Coming to another type of secondary structure, which is the beta conformation. So beta conformations have a structure which is completely different from alpha helix. Here it is not wound in a helix, but it is completely extended. So the backbone of the polypeptide chain is extended into a zigzag structure rather than a helical structure. So these are much more longer in their structures. The beta conformations is much more longer as compared to a alpha helix. And the polypeptide chains, which are in the zigzag form, they can be arranged side by side to form a structure which resembles a series of pleats. And this entire structure, with like many of the polypeptide chains in a zigzag form which are formed, that entire structure is called as a beta sheet. And again, the beta sheet structure, like the alpha helix, is stabilized by hydrogen bonds. So these hydrogen bonds are formed between adjacent segments of a polypeptide chain. So the individual segments that form a sheet are usually nearby, but they can be quite distant from each other also. And we can have both a parallel orientation or an anti-parallel orientation for the polypeptides. The R groups of the amino acids usually protrude outwards, like as in the helical structure of alpha helix, where the R groups are outwards. Here also the R groups protrude outward from the zigzag structure to avoid any kind of steric hindrance. So if we look at this particular structure over here, you can see that this is the extended polypeptide chain which is in the zigzag form, and you can see that there are, when many such polypeptides join together, they will form what we call as a beta pleated sheet, and the R groups are all protruding outwards from this beta pleated sheet. You can also see that there is this hydrogen bonding between, again, the hydrogen of the amino group and the oxygen of the carbonyl group. They are hydrogen bonded, and these hydrogen bonds again stabilize the structure of the beta sheet. The R groups can also interact with each other to further stabilize the structure of the beta sheet. So here again, a similar structure you can see, which gives us an idea of how these are in a zigzag form and how they form what we call as a secondary structure, which is the beta pleated sheet. So here we can see a comparison of both the secondary structures of the proteins. We have the alpha helix, which is usually represented like a helical structure, and we have the beta pleated sheet. So this beta pleated sheet basically gives us the orientation. An arrow in this direction basically means the direction of the beta pleated sheet is in the these two are in the anti-parallel orientations. So here we can see this is how the beta pleated sheet looks, and this is how an alpha helix looks. So both of them are extremely different structures, but the commonalities between both the secondary structures of protein is that they are highly stabilized by mainly by hydrogen bonding, and then further by other non-covalent interactions which occur between the R groups. And this is how a combination of the secondary structure looks like. So a globular protein will have both alpha helices. So the ones in red that can be seen over here is an alpha helix, and the one in yellow with the arrow orientation is basically the beta pleated sheet. So we can see that there are these four polypeptide chains which are folded into the beta pleated sheet, and these four segments in the entire protein which are folded into the alpha helix. And all of them come together to form the three-dimensional structure of the polypeptide chain. So this entire structure can be called as the tertiary structure of the protein, because this gives us information regarding the association of the alpha helix and the beta sheets with each other. Another structure that we can see over here are the ones in green, which are known as the beta turns. So we'll be talking about beta turns also. So before that, so a combination of alpha helices, beta sheets, and beta turns together forms the tertiary structure of the protein. So if we talk about beta turns, they are basically for linking successive runs of alpha helices or beta conformations with each other. And the structure is a 180 turn, which involves 4 amino acid residues. And in this, the first amino acid and the fourth amino acid usually are hydrogen bonded for stability of the structure. The peptide bonds of the two central residues do not participate in the hydrogen bonding, and they are usually or in any other interresidue hydrogen bonding. Now, the two amino acids which were excluded from alpha helices are find their place in beta turns for the same reason. Glycine, being highly flexible, will allow the 180-degree turn. Proline, being because of its amino nitrogen, it tends to assume a cis configuration, because of which it is easier for a polypeptide chain to take a turn. So in a beta turn, we will usually find glycine and proline. And this is how a beta turn looks. So we can see that there are 4 amino acids involved, and the first and the fourth are usually hydrogen bonded, and this is how the beta turn looks. So these are the secondary structures which are present in the protein. Thank you.